All right, I know we still have some folks trickling in, but let's get started because we have three great presenters, and I want to make sure we have some time for audience participation. First and foremost, I appreciate everybody getting up bright and early. I'm guessing there's some Europeans and East Coasters here, like everybody else was up at 5 a.m. So thanks for joining. Please grab some breakfast. We're really excited at ScaleReady to sponsor this session. Our goal with this session, and I'll be brief so we can get to our presenters, but we are working to redefine sustainability in cell and gene therapy. What do we mean by that? At ScaleReady and all of my presenters up here, we are trying to make the business of cell therapy practical and viable. We really want to ensure patient affordability and accessibility. There's some different strategies to do that, but all of them center around using proven technology that's in commercial products today and building off from that success. So my name is Josh Ludwig. I'm the global director here at ScaleReady, and I've been with the company since we launched in 2021. Excited to continue to build our network, build this consortium of like-minded technology providers to ensure that we have scalable production available. My background, I've been in the space for about 11 years now in cell therapy, and for a handful of years before that in bioproduction. So my background is definitely manufacturing. So this is near and dear to my heart, and how can we create a sustainable future where we get access to more patients? So for those that don't know, ScaleReady, we did launch in 2021. We started with 3 industry powerhouse partners, basically building around core technology. Again, all of these technologies are used in existing commercialized cell therapy products today. We've built it around the G-Rex static bioreactor, of course, putting in the top quality GMP reagents from Bio-Techne, novel gene editing, non-viral gene editing, TC Buster. And centered around this on the outside is going to be our global leader in cell processing from Fresenius Kabi. We have both the Q and Lovo system at the booth, so please stop by, talk to our experts. By bringing these companies together, we really wanted to show the field, if you create this modular system, it's flexible, it's simple, it's scalable, and again, our 3 panelists here are going to show how different strategies can be deployed to use these technologies to create scalable systems. So ScaleReady, we are process optimization experts. We have a team in Europe, a team in the US, but we support our clients globally. So we have these gold standard products that are, again, used in existing commercialized CAR T drugs, other therapies, other T cells as well. And our main focus, again, is to simplify, standardize, and then we scale. And you're also going to see how we're automating here into the future. So our goals really are the same as everyone in this audience. We're trying to deliver more access to more patients, deliver more of these life-saving drugs. We have all this great data. We know manufacturing is a challenge. That's why we're here to simplify. And we do all this without compromising the top cell quality, biology, and really unmatched consistency and repeatability in this space. So we have our 3 speakers here. I have Carol Houts, the Chief Strategy Officer at Germfree, Edwin Stone, the CEO of Cellular Origins, and Ignacio Nunez, who is the Chief Operating Officer at CellReady. They are all going to present. I'm going to wrap up here shortly so we can get through all of their presentations, and I'm eager to have audience participation. So without further delay, it's my honor to introduce our first speaker, Carol Houts. She is the Chief Strategy Officer at Germfree. A little background on Carol: she brings nearly 3 decades of experience in pharmaceutical and biologics product development, quality, regulatory affairs, and clinical research. So her strategic acumen has been a driving force for all you're doing at Germfree. Carol's also known for enhancing organizational capabilities and driving revenue growth. She excels in business development and strategic planning, and at Germfree, she'll continue to use her industry knowledge to propel the company to success and as they design additional facility solutions. So please join me in welcoming Carol.
Good morning. So first I thought we'd start out with, well, what do we mean when it comes to sustainability? And I'm going to talk specifically about sustainability utilizing a decentralized manufacturing model. So for us, we're really looking at patient access. How can we move the needle on affordability so that we, through operational efficiency, so that we can bring more of these therapies to patients? We know from a cost perspective, we're still in a place of high cost. We have a number of drugs on the market today, which I think has been attributed to a lot of folks here in the audience. But as you can see, we're still at a price point where it's really unaffordable and unattainable for most patients. And if you look at the cost of the products themselves, $375,000 upwards is not really sustainable in the future. And if you're looking at the therapeutic cost, the overall treatment, we're at like $600,000 to $1 million. So we know that for most healthcare systems, this is not a long-term solution. So what we believe is the future of decentralized manufacturing will help gain us some efficiencies. And the way we're looking at this at Germfree is really that we're developing technology in 3 phases. And a lot of this technology is available to us today. And I think if you're thinking about where we are, there's a lot we can do to move the needle on patient access today. So if you're looking at centralized manufacturing is really where we are today, and I'm calling that 1.0. There's a lot of complexities around the current infrastructure when it comes to efficiencies around cryopreservation and logistics. So as I look towards decentralized manufacturing 2.0, there's some technology that Germfree has developed over the years that really falls into this category. And I think that we can really do a lot on cost reduction in this phase. And I do think that digital systems is a key component. And when I mean digital systems at a 2.0 stage, it's really all around controlling process. And of course, the quality release piece of that is intricately important. So decentralized manufacturing 3.0, I really look at this as where we're bringing in some automation. And you can kind of see here what I'm thinking about for timeline. And I think this really needs to be debated. So maybe in 3 to 5 years, our cost target could be around maybe $150,000. I'd like to see it drawn even lower than that. I think we can achieve that. And then maybe that's in 3 to 5 years. So maybe some of the commercial products will be at this stage going forward. And then I do think, and it's bold, that maybe decentralized manufacturing 4.0 is, could this be delivered through specialty pharmacy organizations? Could this be delivered through our blood systems today? Why not? Some of the most complicated drugs on the market today are compounded by a pharmacist before they're given to a patient. So if we're truly going to scale, maybe this is ultimately the model. So why does decentralization make sense? We know about the complexities of logistics. We know that some of the recent conversations at the advisory board recently at the FDA, we're still looking at that 35 to 50 plus days for the manufacturing process. In that time, patients are not receiving positive therapy. So we've got to move the needle on that. Patient outcomes are certainly affected. So operational efficiencies, reducing infrastructure logistics, the complexities, enhancing the ability to scale and flexibility. And we really believe that with existing technology, and I think the G-Rex system plays a big, important part in this. It's commercially approved. Products are on the market with it. I don't think at this point in time we should reinvent the wheel. I think we should utilize what we have, what was proven, and then move forward and be bold when it comes to decentralization. I think there's a lot of innovation, especially on the quality and quality system side of this. So just a little bit about Germfree. So I'd say that if I was to describe Germfree, we're really a platform technology manufacturer. So on the facility side, we provide both modular and mobile clean rooms, and then we provide a lot of equipment and outfitting. And I'd say that the future for Germfree is really going to be all about providing manufacturing solutions that clients can step into and then ultimately manufacture these products. So a little bit of a timeline here. So back in 2014, 2015, our owner at the time had this incredible idea. Hospitals were starting to renovate to comply with USP 797-800. It was a massive shift in the industry. And at that point in time, he had this crazy idea, let's put a clean room in a mobile 53-foot trailer. And I personally thought he was crazy until I saw it myself. And over the years, we've deployed more than 110 clean rooms around the United States. We have a fleet of about 20 units that are out in the field, operating at hospitals or at pharmaceutical companies, producing GMP or producing compounded medicines compliant to USP 797. So if you look at around 2015 to 2017 is when we really started first deploying units. And we also at that time were looking at the business model. We knew that capital constraints were really going to prevent some hospitals from implementing this as a solution. They were already investing in facilities. So we actually came up with leasing models and managing units remotely. We built a service team to support that. And then if you start to look at, in 2020, of course, COVID hits us, we start really getting in earnest into the GMP world at that stage, doing some work with companies to launch clinical trials, and then also started really looking at decentralized manufacturing with some of the projects around vaccine production back then. And I'd say now in 2023, in 2023, we deployed a couple of units for viral vector manufacturing at a point of care facility, a full-on NCI accredited facility in Florida. And then very interestingly, last year, we did some work with a company called On Demand Pharma. And unfortunately, in the United States, you have two major challenges that Germfree is tackling. Of course, one is all around patient access to cell and gene therapy products. The second one is all around drug shortages. And I've been in the industry more than 30 years now, and I find it shocking that drugs that were approved in the 80s are not available to patients today, that if I have cancer, first-line therapy might not be available to me simply because it's not available. Those products have been offshored from a manufacturing perspective. So we really worked with On Demand Pharma to develop a decentralized manufacturing model to address drug shortages. And then in this year, in 2024, we're actually developing and will be manufacturing kind of the first decentralized manufacturing units intended for cell and gene therapy, where multiple sites will be linked together to participate in manufacturing and ultimately clinical trials. So we really, as a company, are working to partner with therapeutic developers. And some of the work that we are doing with Caring Cross, everyone kind of knows some of the work that they're doing. It's very important work when it comes to our mission and sustainability and getting patient access and being able to scale these therapies. We've also doing some partnerships with Orgenesis. We are very responsible for Biotainer. You may be familiar with some of the work that BioNTech is doing in Rwanda, and of course, On Demand Pharma. So from a readiness perspective, this slide probably is familiar to many of you. It takes a lot of thought and process up front when you start to look at decentralization and looking at every pain point along the process and every control point, and it's got to be something that's considered well up front in the process. When it comes to quality systems and oversight, I think from a regulatory perspective, we've all settled on the model of central site oversight, being the IND holder and the BLA holder, same process in Europe, I'd say. Also with QP oversight when you start to think about this in Europe and other regions of the world. The way we see it is kind of two scenarios, a point of care manufacturing site, and then I do think we'll see regional decentralized manufacturing sites that then also work closely with patient care sites around the world. So some of the work that we're doing with Caring Cross and Fiocruz in Brazil, we couldn't be more excited about this work. It's very important work when we look at how we will address this important issue globally. So Caring Cross will be doing some tech transfer work and training Fiocruz on on CAR T cell processing. And they will pilot the process in a manufacturing unit that we're actually in development now, and in collaboration also with Ohio State. So in the first instance, now we have sites collaborating around the world to actually produce CAR T. So it's super, super exciting. And these units will be eventually scaled. As some of you might know, Brazil has a constitutional requirement to provide advanced medicine, so this is a very big deal. If you can imagine at the current price point, that would break a healthcare system like Brazil from a cost perspective. So this is a little bit about their goals. Ultimately, the savings is kind of noted here, and what we think we can achieve in a cost per patient dose. And it can't be underscored the importance of this work and how we can look at this globally. So obviously looking at streamlining process has been very important. Utilizing commercially available products like the G-Rex, I think is a critical part of the strategy. There's a lot of complexities around decentralized manufacturing. I think going in with proven technology is the way we'll move the needle. Collaborative best practices, I think bringing large hospital centers and organizations that have really developed capability for these first early phases, it's going to be very important. In Fiocruz, I'm not sure how many people know, they produce a lot of the medicines, especially vaccines, for Latin America in general. Simplified QC, I think a lot of amazing work is being done on this front. And then obviously looking at digital systems, we're looking at some even all the way back to where the healthcare system is involved in actually delivering the therapy. So one of the things that was very important is really creating a uniform facility design, like for like copy. This is not an identical unit. The FDA, in my conversations with them, have said, do not say it's identical. So these are like for like or comparable units, and we're working very much in partnership with them and getting their advice all along the way and other regulators around the world. So our intention is to standardize qualification protocols and comparability assessments. I think this is also where standard equipment and processes are really important, especially those that are commercially proven. So that we can reduce some of the regulatory complexity. We got enough challenges ahead of us on the quality system side to move forward. So standardization is going to be key to the comparability, and then obviously cost reduction. So this is a little bit of a case study on on-demand pharma. So this was something that we first dosed patients last year in December. Really exciting that we've been able to produce more than 6,000 doses in about 3 months. These were all shorted drugs. So the hospitals have an endless amount of resources they spend to try to source shorted drugs. It's incredible. So the fact that they could produce them on site was really impressive. And you can see from the picture, we did lots of process scenarios and walkthroughs to get us there. So, and this directly correlates to what we're trying to do in cell therapy. So you can see from an oversight perspective, a pharmacist is licensed in the state of Mississippi. And that pharmacist has to oversee everything that's going on in that clean room. So you can see that she is there looking at essentially the equivalent of a batch record. She's got cameras in the unit. She's got cameras in the biosafety cabinet, in the laminar flow unit. She can see how the pharmacy techs are operating. And then you can see in Rockville, Maryland, the team actually overseeing there as well. So their quality team is there, and their head pharmacist as well. So you can see the technology is here. So now it's time to actually move forward, and let's do this for patient access for cell therapy. Orgenesis has done some great work as well in Spain. There's an OMPUL placed there in 2002. This is actually for TIL, so I don't want us to forget about TIL. In the background there, I think there's a lot of great work that can happen there as well, and there's some clinical trials there that's being supported. So what have we learned in this process? So I said more than 110 units we've deployed around the country. So I'd say localization is critical, and we spent some time in Brazil recently looking at sites, how can we get modules in tough places, in urban settings, things like that. Thankfully, the work that we've done from 2014, 2015 to present, we know how complicated healthcare centers are. They don't have abundant space. So you find clean rooms and closets and things like that. So this localization piece is very, very important, looking at materials, logistics, obviously workflows and proximity to hospital, regulatory requirements, engaging with regulatory agencies early. I can't say enough about that. And regulatory agencies doesn't mean just the Ministry of Health. It means local regulations that govern patient care. You also have to consider. So risk assessments, very foundational for us. We like to use the ISP baseline guidelines. So again, using proven standards and regulations and approaches as we start to innovate in a very iterative way. For us, CQV starts the moment contracting starts. So we're thinking about how are we going to complete our validation of the facility right from the get-go. And one of the things that I used to get asked frequently is how many times could you move a mobile unit? And I'd say it's twice per year. When you look at the amount of validation effort, you could only move a clean room twice per year. And then after that, it just wouldn't be attainable. Space is precious. Really giving as much space to the end user is critically important. And I'd say that be careful about engineering controls. You can't overdo it in small spaces. So you want to make sure that your operators can continue to be efficient in their operations. And standardization like for like is really required, and that provides efficiencies in training and how equipment is handled, supply chain. You can imagine when you have like for like units decentralized, how important that really is to make sure that comparability is met, efficiencies are met, and obviously the main driver, patient cost, being foundational. So understanding how your people move through a facility, materials, product, waste, looking at the ways that even when there are failures, how will they address them in a decentralized model is really important. These process simulations, I think, are foundational to make sure that we can expedite and simplify implementations. Remote monitoring for us, it's been something we've been doing for a lot of years, but now, as I showed you for on-demand pharma, that's being elevated now moving into cell therapy. What can we do from a remote monitoring perspective? You can see cameras can help show how operators are producing. We've got quality management systems, electronic batch records, all of that stuff we can do today. Obviously, preventative maintenance and really being ahead of the game when it comes to supporting these manufacturing units couldn't be more important. And also designing and building facility all around longevity and durability of facility solutions. So you can imagine when I showed you that pharmacy where there's more than 6,000 doses that they've had, you can't have patients waiting to receive their chemotherapy and tell them, no, sorry, your clean room is down. So you have to have nearly failure-proof design on these facilities to make that attainable. So we always have to look at clean room cleaning. These are kind of tight spaces. How do you go about that? And obviously, getting a robust process in place, rolling that out to standard sites is really important, and then actually measuring and monitoring that through your EMPQ, and then your ongoing environmental monitoring program is really important. Strong turnover procedures, you can imagine, as if we have a mobile unit coming back to us from the field, Germfree has really innovated on how we go about doing that. It's not something that you can just bring back to the shop, take a look at it, send it back out. We've got decontamination technology that we've developed over time. We use a lot of VHP in our processes, and I think these are tools that we'll need going forward. So thank you very much for your time, and I'd love to hear some questions on the panel.
Thank you, Carol. Fantastic talk. Really exciting about the collaboration with Fiocruz and Caring Cross and sustainability and patient access, and it's going to be great. So our next speaker, excited to welcome to the stage Edwin Stone. He is the CEO of Cellular Origins. Edwin is an automation expert. He's been at it in cell therapy for at least 20 years, I believe. And he is going to talk through what they are doing at Cellular Origins and with their Constellation system. Please join me in welcoming Edwin Stone.
Good morning, everyone. Thank you for getting up early. Thank you to Josh and the ScaleReady team for the invite to talk to you this morning. I want to start by just thinking about the current state of manufacturing. These numbers, I'm sure you're familiar with. You've probably got your own versions of these, but these are some public domain numbers from the Kite facility in Amsterdam, where they've now got 19,000 square meters. Apologies for those that speak in square feet. The conversion there is relatively simple. That's a factor of 10, so 190,000 square feet. Or in American football fields, I believe that's around 4. I'm not an expert in the size of American football fields, so we'll move to tennis later for those that speak in Roland Garros. That's not the total sum of the problem, though. Perhaps much harder is staffing of that facility. 900 highly skilled employees, and all of that can only produce about 4,000 therapies a year. And so if we start to think about what that means in terms of output of that facility, at any one time in that facility, we've got 100 doses in flight, which in itself doesn't sound that bad until we think about that's getting 10 doses out a day. If we think about that as a hospital, the idea of a hospital that's got 900 staff is 19,000 square meters that was treating 10 patients a day would be ridiculous. No one would build that facility. Each year, a single employee is only able to produce about 4.5 doses worth. So each day, each employee is producing 1/100th of a dose. And that's taking an area of one tennis court. So at any one time, those 100 doses that are in flight are effectively occupying within that facility a tennis court's worth of space. And that's at 4,000. If we think about the indications we've now got approved and that we've now moved through the early part of this year, if we want to start to go to 20, 40, maybe one day with the larger indications, hundreds of thousands of patients, these facilities are impossible. We cannot build facilities of this size. We cannot staff facilities of this size. And the encouraging thing is we've solved problems like this before. For those people that remember the Amgen EPO line, over 20 years ago now, that had 40,000 batches at any one time in process. Now, the first to admit, this is not entirely back to Carol's like for like. It is a different process, but it shows what intensification looks like. It shows what industrialization looks like. And I think if there's something that I'm really passionate about, I think we need to stop conflating commercialization with industrialization. The fact we've got commercial approval, the fact that we've got reimbursement for a drug, does not necessarily mean we're anywhere near industrializing that. But we can do it. To give a sense of what that means, if you've got 40,000 batches in any one time, that starts to give you a production output of 1.5 million doses a year. That's 375 times more output than the facility we were talking about a second ago. And all of that's done within 6 tennis courts. So you've got a 17 times smaller facility. Again, recognizing this is not a like-for-like comparison, but it gives you an idea that we're not talking about an order of magnitude or 2 improvements. We're talking about 3 orders of magnitude, denser production of some of these. So it gives, I think, great optimism that there is scope to improve on what the current status quo is. And how might we do that? So I think we believe at the heart of a lot of this is looking at how we robotically enable the existing modular processes. As Josh alluded to at the start, we have great technologies now available to us to manufacture cell therapies. The key now is how do we use those efficiently and effectively? Thing number one, we believe, is removing human operators from the processes. Human operators are fundamentally inefficient, particularly at repetitive tasks. Humans are great at innovation. Humans are great at the exceptional. And I think actually using humans deep into the process development and deep into the clinical trials where flexibility is key is really powerful. Using humans to do routine tasks in manufacturing is not a viable route for long-term success. By reducing the number of operators in a facility, we cut the amount of labor, we cut the amount of errors, and we cut the amount of waste. Similarly with capital equipment, what we need to do is focus on building facilities that really expand capacity at the bottlenecks. We have a process that is typically dominated by an expansion piece. That is comfortably the longest part of the process. What we need to do is look to build the facility that has capacity where we have the longest operations and use the earlier parts where we've got systems that where we're taking half a day, we only need 1 or 2 pieces in that part of the process. And all of that means we can reduce footprint. And by reducing footprint, we can have facilities that are now much more efficient from the point of view of emissions, much more efficient from the point of operation. I'll just show you a short video of what the Cellular Origin system looks like in operation. So we have the ability to transport consumables, to move bioreactors, to move devices from one part of the system to another. This is obviously the G-Rex consumable that we're moving, and similarly we can move cell products and cell bags. And we can join these devices together aseptically, so we've automated the challenge of tube welding. And finally, we can automate the process of fluid transfer. So we do this through a sterile pumping system that's able to move fluids in the same way you do at the moment through peristaltic fluid transfer. And once we do that, we can then move whatever the consumable, the bioreactor, or any other device in the process into the incubator. We can take it out when we need to sample from the incubator. And by doing all of this, we can eliminate an enormous amount of the incumbent labor in the process. And so let's look through and see what that actually means in practice. We believe that the right automation can take 90% of the labor out of the process. The things you saw there in that video show you that you've got a huge amount of opportunity to remove labor within the fluid transfer, within the kit loading, unloading, and within the instrument setup and operation. Around 40% of the labor in the process is actually tied up in those steps alone. So by removing that, we're removing almost half of the labor we see in the processing. What we didn't see in the video is the opportunity to remove labor within things like the at-line testing, and absolutely crucially, but almost that comes for free when you're building automation, is the ability to do automatic batch tracking and record retention. So every time we do a fluid transfer, every time we do a consumable transfer, we can record that process and we can store that in the batch record. And that's around 30% of the labor in the process. And finally, it's the things that fall outside of that core expansion system. It's the things that start upstream and come downstream of that, things like the automated media prep, the automated reagent prep. We often neglect that. They're not really visible in what we do, but they're things that occupy about a third of the labor in the process. And so by doing all of that, we end up with only about 5% of the labor remaining. And to give you a sense of what's left there, it is things like equipment service. It's things like some of the kitting processes that are challenging to automate as the world is today. But that's not to say long-term we cannot. And so could we at some point get it down to 1% remaining labor? Absolutely. But of course, we do not need to. If we can reduce the labor by these sorts of amounts, we're already making a massive improvement. We're taking the 9,000 operators down to more like 500s. Just to bring it back to traditional sustainability, we're starting to talk about them being able to save up to 40,000 tons of CO2 per year, which tons of CO2 per year is a little abstract. I'm reliably informed that one Big Mac is about 2.3 kilograms of CO2. So by taking out that 8,500 people, we're saving 18 million burgers worth of CO2 every year. And talking about space, again, looking at that Amgen facility, we can see that space is wasted today. And particularly when we think about all-in-one operations, anywhere where we start to combine inefficiently a day zero process with an expansion process, with a harvest process, there is an inefficient use. Typically, we'll be tying up 1 square meter of factory floor space for the entire length of that 10-day process, 14-day process. It's pretty simple, just thinking about that incubation step of how much smaller something like a G-Rex is compared with an all-in-one solution. For that part of the process alone, even without looking at 3D, even just looking in a 2D space, we can start to save around 1/100th, or we can start to reduce to 1/100th of the space requirement we would be otherwise. And of course, that's the most intense part of the space of the process. But if we look at that across the entire manufacturing process, the work we've done, we believe we can get about 30 times more efficient in space utilization. And when we go back to the Amgen numbers, that's not trying to suggest some sort of radical solution. This is something that's obtainable today. It doesn't require us to develop new biology. This isn't new science we're trying to do here. It's nowhere near on a par with all of the wonderful work that's happening around improvements in CAR-Ts and improvements in the fundamental biology. This is very simple industrialization, exactly the same techniques that have been used in every other industry. That can easily get us to be able to produce 1,000 doses plus in 400 square feet. And back to thinking about what that means, which is very directly applicable, is we all know from our clean room operations of what that means in energy usage and energy cost, but also then in CO2 production from those facilities. So based on that 40,000 a year facility, we're talking about 12,000 tons of CO2 that we could save there. And again, to put that in context, a lot of people have traveled here, I'm sure, by plane. That would be the next slide if we could get this to advance. Thank you. Which is about 12,000 transatlantic flights. So there is enormous opportunity that is readily accessible. And talking about fundamental sustainability of industry, we've spoken a few times now about cost. Cost is a wonderful indicator of waste. As an economically driven society, cost is really kind of our core indication of how efficient we're being in production. As we reduce cost, we inevitably have reduced waste. And so with a manual process, and again, these are numbers that everyone will have slightly different takes on, but this was some work we've done with customers that we are use as a reference point in a lot of the work we do. You're talking around £70,000, touching $100,000 of cost of goods in a manual process today. By automating that, we can reduce 95% of that labor. We can reduce most of that facilities cost as well. And that means we come down to around half of that cost. And with all the associated savings that come along there, and that, to Carol's timeline of where we want to go in the future in terms of cost reductions, is accessible. It doesn't require new biology. It doesn't require us to solve major fundamental scientific challenges. This is implementation of good industrialization. And it is happening now. So, I mean, we're very fortunate that we've got the tools and technologies out there, working with people like ScaleReady, who have got solutions that are highly amenable to efficient scale. You might have seen yesterday, we made an announcement about some work we're doing with the Cell Therapy Catapult to collaborate with them to show these facilities in practice. I'm a great believer that as good as anything is in a theoretical sense, until we've shown it in practice, no one should necessarily believe what we say. We are making these a reality now, and so very soon we'll be able to talk at conferences like this about actually having achieved those sort of reductions, about having achieved a 30% reduction in space, having achieved a 95% reduction in labor, and having achieved a halving in our cost of goods. And I think that's critical to the future of our industry. To the topic of today's discussion, I think that is what we need to achieve a sustainable future for cell therapies. Thank you.
Thank you, Edwin. That was a great talk. Amazing to see driving down to 5% of the labor after you guys get this Constellation program up and running, and really excited about the Catapult effort. Okay, our next speaker is Ignacio Nunez. He is the Chief Operations Officer at CellReady. Before that, Ignacio was the COO at Chiromic, a cell therapy manufacturer. He has many stints at different pharmaceutical companies, Johnson & Johnson, Novartis, helping to implement manufacturing improvements for Kymriah. He is a Lean Six Sigma black belt, and he's going to talk about how he is utilizing those skills at CellReady. Please join me in welcoming Ignacio.
Thank you, Josh, Carol, Edwin, phenomenal presentations. My name is Ignacio. I'm the CEO of CellReady. CellReady is a G-Rex-centric CDMO in Houston, Texas, founded by the owner and co-creator of G-Rex's and Wilson Wolf, John Wilson. Our vision is to structurally reduce the cost of cell therapies by applying standards and by teaching organizations, academia, biotech, to use G-Rex in the right manner. Today, I would like to talk about sustainability and waste and how we can turn that into our competitive advantage. Now, maybe when you're thinking about sustainability, you think primarily on environmental waste. You're talking about paper, you're talking about plastic, you're talking about CO2. When I think about waste, immediately, because I'm a manufacturing guy, my mind goes to the 7 types of waste as are defined in lean manufacturing methodology. So actually, what I wanted to do today originally was to talk to you about the 7 ways as they apply into cell therapy. So I was going to talk about overproduction, defects, overprocessing, motion. But I just realized that the industry is way too green for this. I mean, the industry is really not prepared to apply this level of fine-tuning in terms of continuous improvement. We are way ahead of this. We have not even started with the first or second cycles of continuous improvement to start talking about this. Let's do a test. How many of you have you heard about the 7 types of waste as they are defined in lean manufacturing? Could you raise your hand? Okay, that's 10%. How many of you know about lean manufacturing or Lean Six Sigma as continuous improvement methodologies? Could you raise your hand? Okay, 20, 30%. Okay, that's awesome. But now you know what I mean. So we are not ready. Look, Lean and Six Sigma and continuous improvement methodologies have been around for over 60 years. They started in manufacturing automobiles, and then they moved into other industries. And they are all around the world, including pharma, traditional pharma, traditional biotech, medical devices, or food industry, but it's not in cell therapy just yet. We are far from there, right? So, okay, so we can now talk about this. So if we can now talk about the 7 types of waste, how do we define waste? I said, well, let's take a step back. Let's start with the why. So why do we do this? We do this for the patient. Cell therapies are meant to cure. They are meant to impact patients. So to me, the biggest waste is to have some science, to have awesome technology, and that science, that technology does not reach the patient for the wrong reasons, because you have very wasteful processes, what have you, but that's the waste. You don't save the lives that you anticipated that you could with your science. That, okay, so we see that we cannot reach the patient, so the reason was what, let's try to find the reasons why. So by finding the reasons why we don't reach the patient, we have to start by identify what are the critical success factors of a company. And there are primarily 3 critical attributes that makes a company sexy for investors to get the money that you need to set up your operations and reach patients. So the first one is that you have your science, that you have a strong scientific basis for an unmet need. The second thing is your ability to generate data fast, that you get into the clinic and generate data in an economical manner. The third thing is that you have a manufacturing platform that will take you from preclinical to clinical, bless you, and eventually to your commercial manufacturing. And you don't have a major overhaul in the middle of that. So when you analyze the 3 critical factors, and you highlight really what makes or breaks every one of the 3 attributes, you realize that actually comes back to economics. It's really a financial model. So you have to know your numbers. You have to understand where your money is going to, because you may not make it. So, okay, so let's look under the hood. So let's learn our numbers. So if you look into your manufacturing, there are 3 main buckets that could become very wasteful and altogether catastrophic for your company. You have your overhead, you have your labor, and then you have your material. Let's start with material. Let's say that we're using a G-Rex, and instead of having a yield of 15 to 20 billion cells that you should get with a G-Rex 500, you get 3 billion cells or 4 billion cells. So in order to generate the same amount of cells that you could if you know how to use G-Rex, you need 4 times more resources, 4 times more G-Rexes, 4 times more media, 4 times more reagent, what have you. Second thing, labor. How many C-suite individuals do you need to bring a small biotech from preclinical to clinical? I've seen a lot of biotech with 5 to 10 C-suite individuals, multiple layers of management, with so complex quality system that you need to have an oversized quality organization just to walk your own process that has nothing to do with the FDA. And then the third thing is the overhead. And with the overhead, I'm going to focus on your facility, which is by far the biggest of all. And then the 1 million question that everybody's going to have to ask, do I go internal or I go external? So if I go external, who is or what is the right partner for my company? If I go internal, well, does it make sense from a financial standpoint? What has to be right for me to go there? Or is better a hybrid model? Okay, so I would like to spend a little of attention here and hopefully give you some numbers that are going to help you to identify how wasteful all this process is and when is the right moment to go from external to internal. When Kymriah was launched in the last week of August 2017, back in the day, I already started hearing, look, we have a manufacturing capacity issue in the US. We have 4 or 5 times demand, or the projection is going to have 4 or 5 times demand than the capacity that we have. So I respectfully disagree. I've been hearing that already for 5 years, but I don't think that we have a capacity issue. We have an underutilization issue, because I can tell you honestly that I believe our manufacturing facilities are less than 30% utilized. Less than 30%. Let me give you a for example. Look to your right. So there you have the manufacturing process step of a typical CAR-T, 10 days. You go from day 0 to harvest. So in the bars, you see the processing time. So if you have a normal 10-hour shift, you have day 0 takes you around 6 hours. Harvest is going to take you 8 hours. So day 0 on harvest utilizes your 60, 80% of your time. Phenomenal. What happened in between? Between day 1 and day 9, you have some days 0 utilization. And then some days don't go higher than 20, 30% because you pretty much do some media exchange, you do cell count or something like that, right? This is one of the standards. What happened the rest? Because there we're talking about facility, we're talking about clean room, we're talking about people, we are talking about wave bioreactor. You use a wave bioreactor, you're talking incubator, you're talking centrifuge, you're talking about all those critical assets that are really expensive are utilized less than 30%. Okay, so let's do the following. Let's do a simulation. Let's say that we want to go internal. We want to build a manufacturing facility, right? I'm not going to take into consideration the retrofit cost or even buying the equipment. I'm going to take into consideration just the running cost. And I'm going to include 4 ISO 7 clean rooms. I'm going to include 25 people, personnel. I'm going to include lease, utilities, IT, equipment, maintenance, calibration, validation, all the good stuff. Just keeping the lights on, okay? $425,000. Now, bear with me. I'm using the Houston, Texas standard. If you want to build your manufacturing facility in one of the most expensive places on earth, like San Francisco or Boston, then you may have to add $200,000 on top of it, right? Okay, let's take that as an standard. $425,000. Okay, so now what is the production cost? Do we have a production cost? So what should be the cost of manufacture just one therapy? And actually, we do have a standard because the G-Rex is utilized by over 1,000 companies, is in 4 commercial cell therapies, and is in like 20, 30, 40 clinical trials. So we do have a standard that we know very well. So we have what we call the G-Rex index. And the G-Rex index is a combination between cost and throughput, right? So what is the cost of one G-Rex 500 to process one G-Rex 500? So not the device, is to process the G-Rex 500. And then what should be the throughput of a G-Rex 500? So a G-Rex 500, let's say we go with a CAR-T. The cost of manufacturing should be around $30,000. If you do TILs, should be probably around $35,000. Now, the throughput that you could expect out of a G-Rex 500 is between 15 to 20 billion cells. By the way, we got in CellReady 24.6 already once, right? So even higher than that. That's what you should get. If you don't get that, please come and talk to us. We will help you. No charge, I promise, right? We just want you to utilize the G-Rex in the right manner. If you use a G-Rex 100, then you should get anywhere between 3 to 4 billion per device. Okay, so we have the cost, 30,000 to 35,000, and then we have the throughput. So that is your G-Rex. Okay, so what I'm gonna show you next is a table that is gonna take the theoretical cost of manufacturing 1, 2, 3, 4, 5 patients, and then what is the waste that generates because you're not meeting your minimum demand to break even, right? So we start with $425,000. That is what I had to pay regardless. Let's say that we manufacture one patient for an autologous T-cell, a CAR-T, right? And I said before, $30,000. I put there $60,000 on purpose. Why? Because I know that not very few people are as efficient as CellReady is. So if you go to a CDMO, probably it's going to be like that, potentially higher. So I use $60,000 just to be fair with the market. So if you manufacture one patient a month, theoretically costs you $60,000. But you still have to pay $425,000. That means that you have $385,000 waste. If you manufacture 3 patients, it costs you $180,000. Theoretically, because in actuality, you are paying still $425,000, so you have a waste of $245,000. You know where I'm going with all this? So actually, to break even to a point that it starts making sense to go internal, you have to manufacture 8 patients a month. That is for T-cell autologous. If you go to TILs, TILs are a little more expensive simply because the process is longer. It will be around 70. So 3 patients will be 210, 5 patients, 350. You break even somewhere around 7 patients a month. Now, I also wanted to touch allogenic, because allogenic is a different ballgame, because just with one donor, you may get up to 20 G-Rex. And 20 G-Rex 500 is a massive amount of cells. You could hit 500 billion cells out of just one single donor, right? Now, the cost of those 20 G-Rex will be around $500,000. And then just with one single batch, you will break even. But here is the kicker. You still need around 80 to 100 patients for that batch, because in one batch, if you take, for example, 5 billion cells per dose, you're gonna have doses for 80 to 100 patients. So do you need 80 to 100 patients for an autologous NK cell therapy every single month? Well, it all depends. Know your numbers. But another thing I'm gonna tell you, you pay that regardless, because if you have a hiccup in your clinical trial and you have to start production for whatever reason, you still have to pay the $425,000, right? So here it is. These are your numbers. So before you go internal, it's good that you come out with the G-Rex index, because that is going to take you, or it's going to tell you, hey, should I go internal? B, I can go to multiple CDMOs and ask them, what is your G-Rex index? And then you're going to see which one is really cost effective. So in summary, know your numbers. Every decision add up to your cost. Manufacturing will be your competitive advantage or a risk to your company's success. Use G-Rex index as a way to compare cost, performance, and company health. Manufacturing underutilization is by far the biggest waste. Money matters even when you have patients in your heart. Thank you.
That was fantastic. Thanks, Ignacio. Know your numbers, and CellReady can help you with that, so can ScaleReady, so we would love to. I know we really wanted to get to a discussion and audience Q&A, but I don't want to keep everybody. There's a big day ahead, so we will end here. I encourage you to come visit us at our booths. I know Carol has a booth. ScaleReady has a booth. We'd love to talk, so please set up some time. And really, thanks everybody for joining this morning. Appreciate it. Have a great rest of your day. Thank you.